Is this me? No, this is yours. I thought yours.
All right, so it's with the chance. Yeah, it's
Hi everyone,
actually.
it's Betsy. I'm so sorry that the internet is failing us at this time. Unfortunately, I can't seem to connect reliably to the Zoom with you all, so I'm going to go ahead and ask that Dr. Ann go through the discussion points. I sent the slides to Dr. Ann so that you can reflect on your training activities. And where I wanted to end that was here, highlighting that students can and should lead the world in discovery and research. One message that I always want students to get is that you're never too young or too new to science. You can always come in and make fresh discoveries no matter where you are in your training. And in fact, having creative, open minded individuals like yourselves really makes a huge difference. Let's just remind ourselves that our proposal allows for 10 collection sites. Now, often developing a collection again and refresh our memory. Now recall that in each of those 10 sites, if we had 10 sites, we might have fewer. If we had each of those sites, we would go in and ideally find 3 trees of one species, neem, and if possible, 3 trees of the other species, malea. Understandably, a site may not have all 6 tree individuals. You might have one species or the other, only one or two trees of each species. Completely okay. In science, we often balance the ideal and the real, and we work in the real world to aspire to the ideal, but we also work with the conditions that we have. So we'll have some in a site, some number of trees, ideally both species, ideally 3 and 3, but we'll see what you can find. Then from each of those trees, we would collect 3 leaves, and for our endophyte work, that would be what we would need. But it's possible, as we discussed, that for Dr. Peng and Dr. Ann, more leaves will be needed. So please discuss with Dr. Ann. So you'll have 3 leaves per tree for endophytes, and for each of those, we would cut, surface sterilize, and plate 24 pieces of leaf tissue. Now remember that we talked about putting 8 pieces on a petri dish. I suggest that we use 2 different types of media. This can expand the breadth or diversity of endophytes that you encounter. For example, we might work with malt extract agar and potato dextrose agar. Perhaps we could do 2 plates of MEA with 8 pieces each, and 1 plate of PDA with 8 pieces for each set of 24. So we would have 2 MEA plates and 1 PDA plate for each leaf, repeated 3 times, once for each leaf from a tree, and then repeated again across trees. Then eventually what we'll be able to do is isolate the endophytes of interest and transfer each to 2 slants, one for the University of Nairobi and one for Therefore, for each collection site, what we're proposing is 2 species times 3 trees each times 3 leaves per tree times 2 plates of MEA, i.e. in a given site, if you found all the trees you were looking for, we would need 36 MEA plates. And for PDA, we have 2 species times 3 trees per species, 3 leaves per tree, 1 plate of PDA, so a maximum of 18 PDA plates. And now remember, we can assume that not always will we find both species or 3 individuals per species, so this is a maximum number of plates. Okay? And recall, as we've just said, the proposal allows for 10 sites. We would ideally have such sites in different regions, for example, in the coastal area, the upland area, the rift area. But this can be adjusted totally as you see fit, based on your expertise and knowledge of where the plants occur and what is accessible. Where can you get? Okay. Now I would like to pose 2 challenges that I think can occupy your time going forward as you prepare to be endophyte explorers. So the first is to discuss as a team how many sites will you target, where are they, and what is your schedule. It's okay to have more trees per site if needed, and then fewer sites. Again, we may not have time or energy or access to 10 sites, so even 2, 3, or 4 sites would be wonderful. Remember, geographic differences and environmental differences are super important for answering our core research question. And just to give you an example of how you might want to choose some sites, here's a map of the climate classification across the beautiful and diverse nation of Kenya. And what you can see is areas that are, for example, warm desert climate in red, semi-arid climate in orange, and then moving into tropical savanna climate in pale blue. So this helps us see some of the environmental differences that could lead us perhaps to choose a couple of sites in the red, a couple of sites in the orange, a couple of sites in the blue, and so on. So here's the challenge. As a team, please come up with a proposed sampling scheme, the number of sites, where they are, and a timeline, and make sure that you give yourself enough time to process fresh material between trips, enough time for the endophytes to grow. Okay, so challenge 1. Now, challenge 2 has to do with preparing the supplies that you'll need. So remember, for each site, we need a maximum of 36 MEA plates and a maximum of 18 PDA plates. So please make enough plates for a site before you collect. Extras can then be used for the next site. It's really wise also then to have your Ziploc bags ready to go, a Sharpie to label so you know which tree is which. You can put all the leaves from a single tree in the same bag. So if you had 3 neem trees in a site, you could have 3 Ziploc bags. It's wise to prepare a data sheet before you go into the field. Often in the field, we're tired, hungry, it's kind of hot, we're thirsty, and we're not always thinking as clearly as we should be. It's really easy to forget things. So if you make a data sheet, then it's easy to just go through and record all the information that you need. What kind of information did we talk about? So for example, names, date, and location. Remember that if you have your coordinates, GPS coordinates, or a spot on the map, then we can get climate data and elevation. And ask who is going to be a data recorder, who will go and assess plant height and health, who will evaluate land use conditions, maybe by talking with local people. You've got a lot of potential there. And you can think as scientists, what do you want to know about these sites where you're collecting, and what could help you interpret what your data look like later? Okay, so be creative, and note that endophytes will take a few days to grow. If the weather is really hot and dry, you might find fewer fungi than you anticipate. If that's the case, then please feel free to sample epiphytes as well. Endophytes are inside the leaves, epiphytes are on the leaves, and recall that the epiphytes are often early stage endophytes. They're growing on the leaf surface and getting ready to go into the leaf. So Dr. Ann has experience in sampling epiphytes, and this would be completely fine as an expansion of your study if it's the case that the endophytes are not growing especially well. So challenge 2, as a team, please start preparing your data sheets and plates. Okay. So challenge 1, pick your sites, think about your timeline. Challenge 2, start preparing those data sheets and plates. With those steps, you're in a great position to go forward, and I can't wait to see what's next. Okay, I'll sign off for now and look forward to the next steps. Okay, bye-bye.
All right. I had thought that you send us protocol, safety procedure, do this, take that, make our lives easy, but no, you have to come up with that, which is not a problem. We can always do that, right? So we make our data sheets and the plates.
Do we know where we're going first?
Yeah, we are going to, no, you are going to Kilifi.
Okay.
Yeah, Kilifi is in the coastal Kenya, at the coast of the Indian Ocean. So you expect to find neem and I think melia there, lots of them, but neem I'm pretty sure you find lots of neem. If you don't find melia, it's okay. Don't be surprised. You'll find them somewhere in the central part of the country because they do well mostly in cool areas. Kilifi is hot, yeah, really hot at this time of the year. So
In Kobe, the only place we're going.
there are 2 regions. I think within Kilifi, Kilifi is like the larger name. But now within Kilifi, there are some specific areas we'll be going to. At this point, I'm not privileged to have that information, unfortunately. Yeah.
Wow, recommendation.
Yeah. You can still come up with a data sheet already, say Kilifi, and then maybe leave spaces for specifics, but then the information below how old is the plant, for example, what do you use this plant for, like a questionnaire of sorts, or even if it's not a questionnaire, but some information you want to capture about the plant. At the back of my head, age of the plant is important because again, you might compare a plant that is 20 years old versus a plant that is 10 years old. The endophyte distribution and diversity might be different because of that. Is the plant, was it planted or did it grow naturally on its own? That one can also be uh, part of your data sheet, and many other nice questions that you come up with. Okay, so we've been learning about endophytes from when we start. Do you want to look at the functional roles of endophytes? And basically, this should be somehow repeating itself because by now you know the importance of endophytes in plant production and crop production and all that. So, of course, nitrogen fixation and nutrient acquisition. So this is one very important reason why plants form that symbiotic relationship with endophytes. And when you talk about nitrogen fixation, you're talking about the rhizobium bacteria or the bradyrhizobium, which form the symbiotic relationship with legumes in the root nodules. Of course, as someone answered yesterday, um, endophytes do not have what we call chlorophyll and chromoplast. Therefore, they cannot make their own food. Therefore, they depend on the plant for food. And by food, basically, we mean source of energy or carbohydrates. Yeah. Yeah. Oh. And then, as also someone answered very accurately yesterday, from the plants they obtain shelter, yes, and also uh, nutrition or carbohydrates, because again, photosynthesis majorly makes carbohydrates or glucose. So that is the relationship they have with the host plant. And besides nitrogen, other endophytes can also help plants acquire phosphorus. And remember, nitrogen exists in the air as N2, just nitrogen gas, elemental nitrogen N2, which forms about 78% of the air in the atmosphere. In that form, as much as plants really need nitrogen, they cannot absorb it or they cannot take it. So the plants only absorb nitrogen in the form of nitrates. NO3 negative, yeah? So elemental nitrogen plus oxygen. So endophytes have this unique ability, or rhizobium have this unique ability of fixing atmospheric nitrogen into nitrates, which now plants can absorb. And nitrogen are very important building blocks in proteins. All right, the arginine, all the proteins have nitrogen as their main building block. Of course, we also have carbon, but for proteins, it's so much nitrogen. So it's very, very important for the plant and also for the leguminous plants because these are protein living plants. See, all legumes provide us with proteins, beans, peas, which other green grams, lentils. So for those who are vegetarian, and even if you're not a vegetarian, at some point, sometimes you consume legumes if maybe you're into healthy eating. And so it's very, very important that these plants can take up nitrogen, and the only way they can take it is if it is converted into nitrates. So for these plants, it helps because now you don't even have to depend on synthetic fertilizers anymore. They can just the bacteria can fix the nitrogen, the plant will take up the nitrogen, and after that, the season is complete. The soils are also fortified because now the soils have extra nitrogen from the previous planting season. And if you're into agriculture, or if maybe because biology and agriculture are also closely related, if you might get to agriculture, when we're talking about crop rotation, after you plant maybe beans, this season, the next season you plant corn or maize or wheat. So you rotate them like that between normal carbohydrate plants and leguminous plants to ensure that you have a good balance of nitrogen in the soil. And if you look at the nitrogen cycle, at some point you'll be taught this in your biology or nitrogen cycle. So plants will only absorb nitrates. We have so the atmospheric nitrogen is first of all fixed into ammonia by the endophytes, and then from ammonia to nitrites, and then to nitrates, and then the plants can absorb nitrogen. And for them to sometimes fix this nitrogen, they need some energy from lightning or from light, but not necessarily important. They can still convert, they can still use just the normal sun energy to convert that. But lightning, when lightning strikes, then uh, the endophytic bacteria normally use that energy to fix or to convert elemental or atmospheric nitrogen into ammonia, then into nitrites, then nitrates, and then the plants can absorb the nitrates and make beans and peas that you like or that you consume. All right, another example is azospirillum and maize or corn. We call corn here maize. In US it's called corn most of the time, but we prefer maize. And actually in Kenya it is the staple food. We really consume a lot of maize. We make something called ugali, or maybe in some places it's called pap. You mill the maize and make it into a white paste, or in some places it's called Olienta, depending on where you come from. And then that one now is used. It's actually tasteless, or it can be a little sweet if the corn was, but basically it's normally tasteless. And then now the beef or the soup is what now will give it the taste. So based on how you cook your vegetables of the beef, and then that is what you consume. I know in the US the staple is potatoes, I guess. What's your staple food in the, or it depends with the region, because as the US is also very big.
In the Midwest, it's like potatoes.
And
then like it's also corn and
corn, really?
yeah, we grow a lot of corn there.
Do you consume corn like how do you consume it? Boiled corn?
Yeah.
Or what we call the baby corn, the small ones?
Yeah.
Sweet corn. It's called sweet corn.
Most of our corn is sweet.
Okay.
Like really, really sweet.
Alright. Just say it's okay. Alright. So for corn, maize, the endophyte in question is called azospirillum, and then it forms a relationship with the roots. And as I told you, all plants around their roots, they're surrounded by a mixture of fungi, which is called mycorrhiza. So inside the mycorrhiza, we have azospirillum, and the mycorrhiza is basically fungal mycelia, which have one end inside the root tissue and another end outside the root tissue. So the fungus have that unique ability to absorb phosphorus, to absorb nitrogen, and once inside the fungus, inside the plant, then it can be released to the plant. So basically, they just help the plant to absorb nutrients. In addition, for azospirillum, they also produce a growth promoting substance known as the uh, indole acetic acid, which stimulates root development and improves plant vigor. So without the endophyte, without that acid, then the roots of the plant might be retarded, or they might not grow as required, and then the plant will not be healthy. So again, I know we've mentioned this over and over again about endophytes and the plant health, but these are now real examples on how these interactions occur or happen. So if you go into a field of corn and isolate the roots, or if you induce the seed to germinate, as you can see in that picture, inside that solution down there are millions and millions of spores of azospirillum, and the substance they produce is the indole 3 acetic acid. So again, if this acid is important, actually acetic acid, this can be a means of also maybe producing acetic acid using this. But of course, the vinegar method is much cheaper. And then endophytes are also known to produce a wide array of secondary metabolites and bioactive compounds. And these compounds often possess medicinal, agricultural, and industrial potential. Now, it's now upon one of us or all of us now to find that medicinal or agricultural or industrial potential property from one of the endophytes that you are likely to isolate. So endophytic fungi, for instance, produce compounds like alkaloids, terpenoids, phenolics, and these ones exhibit antimicrobial, anti-cancer, and anti-insecticidal properties. And as the world is moving from chemical to now more biological, more organic means of doing things, these are alternative sources of, for example, antibiotics or, for example, insecticidal compounds that might be required. Instead of having, there's a pesticide that, for example, that you spray on maybe bugs. Uh, it's called Doom. Do you have in the US the biggest brand of pesticide is called Raid. Bagel. What called? No, Raid. Raid, yeah. Oh yeah, this Raid, yeah. For example, Raid. Raid is made from extracts, chemical extracts of pyrethrin. So in future, instead of spraying Raid, you might spray spores of a certain endophyte that will also kill the insects in a very environmental friendly way. Because after spraying Raid, the pyrethrin in it will also affect you. You'll feel that if you inhale it, you feel it's not comfortable. Yeah, yeah. Yeah. So in future, we can use endophytes, and of course, if it is not pathogenic to humans, the spores will do nothing to you. So just like maybe inhaling dust and then you cough it out, or it's clogged in the mucus and you eliminate it. So those are some of the alternative methods or approaches to life that can be explored through endophytes. And a very good example is a compound called Taxol that is produced by Taxomyces andrenae. Taxomyces is isolated from the bark of the Pacific yew tree. So it's an endophyte of the Pacific yew tree, and Taxol is a compound with significant anti-cancer properties. Actually, it has been commercialized, and we have anti-cancer medicine known as Taxol from the endophyte known as Taxomyces. So it is possible to get something from neem or something from Melia. Very possible. So okay, endophytes also contribute to plant growth promoting substances, and they produce plant growth promoting substances like auxins, cytokinins, gibberellins, ethylene, and many others. If you remember your biology, auxins, cytokinins, gibberellins are classified as plant hormones. Yeah, these are plant hormones. Plants also have hormones, not only humans. Of course, humans we know of estrogen, testosterone, and many others that we produce. Those two are the most common. Plants also produce hormones. There is ongoing discourse on whether these hormones are actually produced by the plant itself or they're produced by the endophytes that inhabit the plant. In some classes, you'll be taught that these ones, auxins, cytokinins, gibberellins, are plant hormones. But endophytes from plants have been isolated and have been found to have the ability to produce some of these compounds. And right now, some of these compounds can be exploited commercially. For example, do we grow bananas in the US? Mm-hmm. You grow?
I think.
Okay, not. Yes and no.
South? Yeah, South. Yeah. Like Florida.
Okay, so what happens is that if you don't grow bananas, but we all consume bananas. Mm-hmm. Sometimes if you go to buy bananas, you buy them from the shop when they are still very green. Mm-hmm. And then you go and keep them somewhere for some time, 1 week, and then they turn into yellow, that deep yellow, and maybe they start having they begin to produce some aroma. The hormone involved, or the compound involved in making that banana turn, is what is called ethylene. All right, under fruit development. If you can see that small figure, you can see the banana there. Under fruit development, ethylene is the one that is responsible for ripening of fruits. Now, this ethylene can be used in two ways. One, you can either choose to apply as basic acid on your fruits to increase what is called the shelf life. rather than apply nitrites and those chemicals that are used to preserve fruits. And also, if you are to import bananas from, you say Mexico, or maybe a different country, or maybe even if it's overseas, it will take time. So you want it to arrive at the destination when it is still fresh and it has not gone bad. So some of these compounds are normally used. So when during transportation, you spray a lot of aspartic acid on it, and then it will thwart or it will prevent actions of ethylene. Upon arrival, and now you have taken the shelf, you apply ethylene. Then after 1 week, your bananas are ready. So of course, right now on a commercial scale, they don't use ethylene and aspartic acid. They use certain chemicals to induce ripening or to preserve the shelf life. But as the world is changing, these are some of the things that can be explored. And endophytes have been found to produce these compounds. Of course, there are compounds that are also found in plants. So some plants also can produce them. So maybe on a commercial scale, rather than have plants produce your auxins and cytokinins, endophytes can be harnessed in a commercial setup, and the same way we use microorganisms to produce insulin or growth hormones for human use, we can also use them to produce plant promoting growth substances like auxins or plant hormones for different uses. Improve the shelf life, maybe ripen your fruit, and even if it's the plant in the field, if you want your plants to grow faster, be big, instead of spraying foliars that have uh, artificial fertilizers, you can be spraying hormones. Gibberellins and cytokinins will help them with quick growth and quick maturity. Just the same way, sometimes humans can also use hormones to achieve different desires and different goals we have. We can also exploit that and use hormones in plants to make them grow faster, grow better, and produce more. And some of these can be directly synthesized by endophytes. An example is Pariformospora indica. And I'm sure maybe some of you isolated this from the word indica. What else is called indica? What is the scientific name of neem?
As an actor.
Aha.
Indica.
Yes. So this one you are likely to isolate this. I'm sure it's one of the things you one of the methods you even worked with yesterday. This Pariformospora indica. So it's an endophytic fungus that is known for plant promoting abilities. The indica normally it helps the plant to tolerate stress such as drought and salinity. From the coastal region, from Kilifi, you are likely to isolate this particular fungus because that is a it's a semi-arid area, the climatic conditions there. So it must have lots of this indica to help it uh, sustain itself in those stress environmental conditions. And also, I was telling you, much more research your research can just be, how does endophytes help these plants tolerate stress? What does it do? Because, sorry, for something like drought, it is lack of water, right? But you're being told that an endophyte will help a plant overcome lack of water. What does it do? Of course, it does not produce water, but what does it do? So that one is a question that you can choose to answer in your line of research and unlock. Maybe it may be something very simple that can unlock production of plants in other areas, semi-arid areas. Like right now, if you have a solution for Middle Eastern countries to produce food, for example, if you present a proposal to the United Arab Emirates that in their desert, because they are net importers of food, they don't grow any food. They import food from Africa, Asia, Brazil. So if you present a solution to them that through this endophyte they can be able to grow their own food, then if you are in need for the money, then that's the jackpot right there. So these are some of the other areas of research you might explore. And of course, this one can be part of the things you do as you proceed, maybe part of your PhD project, masters, and whatnot. So for me, I'm curious to find out. Okay, I'm sure endophytes help this plant tolerate stress. and drought. For salinity, it is simple because for salinity, it is just changing the osmotic pressure, osmotic potential internally by either accumulating salts or releasing salts. So for salinity, there's a very good, easy scientific explanation. But for drought, how do endophytes help plants withhold or withstand drought? Think about it. All right. Another very important aspect: endophytes in bioremediation and photoremediation. So basically, bioremediation is the use of living organisms to degrade or detoxify pollutants that have accumulated in our environment. So when you use living organisms to remove pollutants in the environment, that is bioremediation. Phytoremediation or phytoremediation is the use of plants. It is still a branch of bioremediation, but now when we talk about phyto, normally when you hear phyto, then that is plant. So phytoremediation is the use of plants to remove contaminants from the environment. And you all know about things like oil spills. You all know things like maybe like right now accumulation of plastics in the oceans. So these are things that require bioremediation from time to time. And it has been found that certain endophytes possess certain enzymes with the ability to degrade pollutants or promote metal accumulation within plants. So if it's a phytoremediation, the endophyte in the plant can increase the ability of the plant to take up mercury, lead, and the heavy metals in the environment. So along the river, maybe close to a company that deals with mercury and lead, they grow thousands of these plants, and then the affluents they release are free of heavy metals. All the heavy metals have been accumulated in the plants that they grow. So these are some other areas that might be interesting in research, can have very industrial and commercial viability in terms of endophytes, the use of endophytes in bioremediation. And a good example is Pseudomonas and Bacillus. These are bacteria that have the ability to break down the heavy or the long hydrocarbon chains in petroleum, and therefore they can be used to bioremediate areas where we have oil spills. And oil spills are common. Of course, even in the US, we had a case a few years ago where there is a large oil spill in the ocean and fish were dying, and we don't want that. So actually, they used bacillus and pseudomonas to try and remediate those places of the sea where we had the spillage. And this is because these microorganisms have the ability, they have the necessary enzymes required to break the heavy and the long hydrocarbon chains that form petroleum. There's a lot of research currently going on some microorganisms that can break down plastics. Because once we get that, then again, we will solve this problem of plastic waste in the environment. So there is that also, and again, this can be a very good line of research for yourself if you're interested. Endophytes with the ability to bioremediate or to break down plastics. So it can be either you can look at it from the angle of view of photo remediation, whereby you plant a plant with a certain endophyte in it that can either enable the plant to accumulate heavy metals and uptake toxic substances from the environment. Or you go straight to the endophyte itself with the abilities to break down the plastics and the metals, isolate the specific enzymes, and then commercially produce these enzymes so that these enzymes can be used for bioremediation and cleaning up of the environment. Right now, there's a lot going on in the same. Like in Kenya, there are some projects whereby we're trying to use, okay, this is not endophyte, but black soldier fly, if you've heard of black soldier fly, to try and remediate or to try and detoxify waste, actually human waste, poop, that is, and waste from abattoirs or slaughterhouses, and just normal organic waste from homesteads. So black soldier fly breaks down this waste. The larva of the black soldier fly can be used as animal feed, and whatever has been broken down from the black soldier fly can be used now as fertilizer. And this was occasioned by the fact that we previously relied on a lot of we relied on Ukraine. Kenya imported a lot of fertilizer from Ukraine, but since the war, We cannot import fertilizer from them anymore. Perfectly understandable. So we have to find solutions for this problem. So one research that was slightly involved was that of using black soldier fly to try and bioremediate the environment. So part of that is, are there microorganisms in this fly that help it survive this extremely toxic waste so that they can consume it? What is the gut? What is the composition of the microbials in the gut of this fly? But that is not now endophyte, for it's now microflora in insects. So, but it is somehow related to this. All right, that is enough about roles of endophytes. We've said they are very important in agriculture, in environment, in terms of bioremediation, and in plant growth and plant health. Now, applications of endophytes, of course, it will be more or less similar. They can be applied in agriculture and in crop improvement. And of course, in agriculture, we have looked at the different roles, taking up of nutrients, enabling the plant to sustain stressful environmental conditions, and also increase the tolerance of these plants to pathogens. And then endophytes will reduce reliance on chemical inputs. And you need also to look at some of the documentaries that sometimes are produced. There is a place, I think it's somewhere in Asia, next where there's this multi-billion company called, I think, Dow, that produces some chemicals. Around that place, people are complaining of certain diseases because of the influence from this company was getting into the ecosystem, into their waterways, and eventually was affecting them. So you see, people will come with unique diseases that have never been diagnosed. So again, when we use chemicals, it's important also for these companies to you also, and I think that is what also Besti mentioned yesterday, that some of this knowledge directly puts you in the driving seat in some of these multinationals, whereby you are a consultant for these companies, but what your role is in terms of bioremediation or the waste that they release into the ecosystem. Already you have ascertained that there are enough endophyte spores that will detoxify it before it goes to the environment. So we're talking about applications of endophytes, and we have said that they're very important in agriculture and crop development. We talk about the hormones or the growth stimulating substances that endophytes can use. Another important aspect of endophytes or use can be sustainable pest management. So endophytes offer sustainable alternatives for pest management because they can act as biological control agents, inhibiting growth and activity of plant pathogens and pests. A very good example is Trichoderma and Bavaria. And as I told you, Trichoderma hasianum today in Kenya has been processed into a compound that can be used to control uh, uh, bacterial wilt in tomato plants or fusarium wilt. So the spores from that plant, the spores are harvested, and then the spores are granulated, and they are also encapsulated using wheat flour mixed with maize flour, and then it is packaged at room temperature. So before you plant your beans, no, not beans, before you plant your tomatoes or tomatoes or potatoes, then you can apply Trichoderma xenum on your field just to ensure that you do not suffer from bacterial wilt. Another one is Bavaria, actually called Bavaria bassiana. It's used a lot in flower farms, whereby that fungi is applied, and when it gets a beetle or an insect, it colonizes the insect, and as you can see from that picture, that is what it does to the insect. And it also normally makes the insect, it also affects the insect becomes like a zombie, because once it colonizes the insect will get the desire to climb to the apex of the grass blade or of a tree, because when it is up there, it will release the spores to a wide area, and then many other insects can be affected. So these are two very good examples that are actually being used today to prevent this one bacterial wilt, this one pest. So these are pesticide. Bavaria is used as a pesticide. Trichoderma is used as a as a fungicide, and there are already products in the market. So there are many avenues you can look at if you are thinking about commercialization of some of these endophytes and maybe making securing the bag from them. Applications in biotechnology, again, enzymes and antibiotics and other compounds can be isolated from endophytes and used in industrial and pharmaceutical industries. As I was talking about bioremediation, you can isolate the enzymes that are normally used to break down some of the toxins in the environment. All right. Can also be used in the textile industry to make those enzymes. We talk about pharmaceuticals, again, also in the cosmetic industry. So it might be interesting to isolate enzymes from a plant like aloe vera or chamomile, and most of the beauty products that ladies like, or most of the beauty products that are there in the shelf today, they will say that it has extracts of aloe vera or chamomile, and it may be interesting to find out why these extracts are good products for skincare, or why they are good antioxidants or why they have moisturizing effects. So again, this can also be a plant of interest for you. Try to find out the endophytes in aloe vera and so future perspectives and challenges in endophyte research. So we've said that endophytes can be applied in agricultural application, that is the biocontrol of plant diseases, pharmaceutical application, that is the discovery of novel bioactive compounds, and in environmental studies, that is bioremediation, plant microbe introduction, plant microbe interactions, and in ecosystem functioning. So one limitation and one challenge is in harnessing the full potential of endophytes, such as host specificity, environmental variability, and scalability issues. But we have seen it is possible to, to achieve that. But one challenge normally when you're dealing with microorganisms are normally things like those environmental variability, scalability, and host specificity. Like if you get an endophyte that can be used to control certain pests in Kenya, can the same endophyte work maybe in the US? It can be yes or it can be no, based on the differences in the environmental conditions between these two localities. Then ethical considerations include potential risk of introducing genetically modified endophytes into the environment, intellectual property rights and access to genetic resources. This is what basically talking about when she mentioned about the Nicaragua agreement, and then compliance with international and national regulations governing biotechnology research. So some of those things are covered in certain agreements like the ones she mentioned. So what areas of research can you venture into? Research gaps for future consideration. So understanding the functional diversity and ecological roles of endophytes. So that one needs to be explored. Or exploring the potential of endophytes in climate change mitigation and adaptation. So right now, everybody is speaking about climate change, and there's a lot of funding towards climate change. So it might be interesting. This can be a very interesting topic, exploring the potential of endophytes in climate change mitigation and adaptation. And maybe one way to do that is to isolate an endophyte from a desert crop and try to incorporate it into an desert crop to make that crop drought resistant. And then developing of innovative methods for endophyte isolation, identification, and characterization. You all know the trouble that you've gone through just to try to identify the endophytes. Already almost 1 week, we are still not sure which species specifically we have. We have to do so for now: isolation, identification, and characterization. There's a method, there's a lab in Bari, Italy that are working on a technique that can be used where you just take, and then it is blasted. And then even before you culture your plant material, you already know the microorganisms that inhabit that plant. So those are some of the methods that can be incorporated, whereby you just take, like when you're in the field, yeah, you take that plant material and you already isolate the DNA from the plant material, you already identify the fungus that is there. So when you come to the lab, you already go and make the media specific for that fungi. Makes work a little bit more easier. So again, you might explore that as a line of research, that one of developing new methods of isolation, identification, and characterization. The good thing is that you've gone through the old school method, so at least from your troubles in the lab, you can now think about how to make it easier. And then investigating mechanisms underlying endophyte mediated plant growth promotion and stress tolerance. Like I was asking you, you are saying that endophytes can help a plant tolerate drought. How? So these are some mechanisms that needs to be studied even more. And basically, yeah, that is it from me for today. All right, thank you very much. And it is yeah, it is 15 minutes to 10, so I think that's good. So we will stop there. I want to thank you very much for your cooperation throughout this week. I believe next week you will be with Professor Sheila and Anne as you go to the field. Maybe you'll see very little of me. Maybe you still see me, we'll figure that out. But I want to say thank you for your cooperation. Yes, but we still be together in the lab. I'm not leaving, yeah? So we still be together in the lab, but yeah. So thank you. So I don't know. It is